I'm very proud to present Emily Caseley from University of Leeds here in the UK. So Emily is currently a postdoc in Leeds. She did her PhD there as well, Wellcome Trust-funded PhD, when she worked on P2X7 receptors, primarily on the ligand binding of these in these receptors. And now during her postdocs, she's focusing on KCNT1 or SLAC channels, which have been directly implicated in certain types of epilepsy. So she, together with John Lippiott and Katie Simmons and Stephen Munch, is developing small molecule inhibitors of these channels, hoping to find presumably new treatments, because as of yet they do not exist for these epilepsies. And this is an industrial collaboration with Connexin Therapeutics. Okay, with that, Emily, I hand it over to you. If you have any questions.
So yeah, thank you so much, Elena, for that wonderful introduction, and thank you all for asking me to speak today. I'm really excited to talk you through some of the work that we've been doing here in Leeds. So what we've been doing is using a structure-based approach to identify novel inhibitors of the KNA1 potassium ion channel. So yeah, let's get on with it. So first up, the KNA1.1 potassium ion channel. So this is the structure of the channel. We're going to be looking at it a lot today, so I hope you enjoy it. So this channel is a tetrameric channel. So it's a complex of 4 KNA1.1 subunits, as Elena mentioned before, also known as SLAC and SLOW 2.2. But in this talk, I'm going to focus on calling it KNA1.1, or sometimes KCNT1, which is the gene that encodes this subunit. Oh, hang on, let me get my pointer. Can we see my laser pointer okay if I have that there? Fabulous, thank you. So we have a potassium selective filter up in this transmembrane region here. And in our intracellular domain, each subunit has 2 regulator of potassium conductance, or RCK domains. And within this intracellular region, we see a series of ion binding sites. So these comprise of sodium, chloride, and zinc, but primarily sodium. So sodium ions bind to these intracellular domains and trigger a conformational change when when we reach a high concentration of intracellular sodium, so in the hundreds of millimolar range. And this causes a conformational change, which leads to an opening of the voltage-sensitive selectivity filter gate and the movement of potassium through the channel. So what does this conformational change look like? So we have 2 structures here, and these are both cryo-EM structures of the chikin receptor. On the left here, we have our apostate, so our closed state. And when we see a increase in intracellular sodium and that sodium binding, we get the conformational change and we get our open state here. So on the left here, you can see that the structure is significantly shorter, about 5 angstroms, than the open state. And these conformational changes facilitate the movement of our ions across the membrane. So this channel has a series of different roles. They're expressed quite widely in the brain, the heart, sensory neurons, and the epithelial cells of the thick ascending limb. And they've been implicated in playing a role in the hyperpolarization of the membrane potential in neurons and other cells that they're expressed in. They play roles in processes including temporal accuracy in brainstem auditory neurons, regulation of neuronal bursting and sensory neurons, and neuronal development. And the reason that we're primarily interested in them is because of their association with disease. So here is yet another lovely structure. You're going to be seeing it a lot, so I hope you're having a nice time with it so far. But within this structure, we can see these residues that are highlighted in red. These are sites of known mutations for KCNT1 associated epilepsies, KCNT1 being the gene that encodes this subunit. So there are over 60 known KCNT1 gain of function missense gene variants, and they're generally heterozygous dominant, and they cause a series of KCNT1 associated epilepsies and associated disorders. So these are all, they vary in severity and age of onset, but generally quite early in life. So age of onset could be from the first weeks through to months of life. these mutations lead to, as well as seizures, as you would expect in a epilepsy associated disease, also developmental delays, cognitive and behavioral abnormalities, and movement disorders. And these can be really severe to the extent where some children who have these mutations, they can't learn to speak or feed themselves. So it's really impactful upon the individual. They're rare, so we are aware of around 3,000 cases, although as awareness raises, the diagnosis might increase, but still pretty rare, and also drug resistant, which is the thing that we're interested in. So Action Medical Research, who funded this work, they produce a story kind of outlining the impact these forms of epilepsy can have on a child's life. So this is Paddy. Paddy is a child who has KCNT1 associated epilepsy, and he can have up to 50 seizures a day. He can't make eye contact with his parents. It's really very severe. And you can see just from that quote with the number of seizures that he has, he's also on a cocktail of epilepsy drugs that aren't really touching any of his symptoms. So if we have any kind of treatment that can be effective for patients like Paddy, then that would have a real world impact. And if you wanted to look at that video, they've got a QR code in the bottom there. It's really worth a watch to find out how these kind of epilepsies can impact a person's life. So what's the epilepsy treatment landscape like at the minute? Well, to be frank, quite bleak, to be honest. There's not much on the market that can really touch this form of epilepsy. We have trials of repurposed drugs. For example, this is quinidine. Quinidine is an antiarrhythmic agent which has been used off-label to try and treat this form of epilepsy. The reason being that we know quinidine is a KNA 1.1 potassium channel blocker. Problem being that there's a myriad of reasons for this treatment not being particularly effective, including poor blood-brain barrier penetration, low potency. So the IC50 of quinidine at the KNA 1.1 potassium ion channel is in the range of 100 micromolar, compared to, say, at hERG is less than 1 micromolar. So by the time you've reached a therapeutic concentration that can have any effect on our KNA 1.1 receptor, the children will have developed long QT syndrome or other cardiac side effects, which mean that they have to be taken off the medication. There have also been other trials. So for example, last year, 2 years ago even, there was a trial with antisense oligonucleotides in 2 children. And unfortunately, that even though in the early stages they seemed promising, the children saw some improvement, they very sadly developed hydrocephaly, and one of the children died before the end of this trial. So this is not to say that these different kinds of multifaceted approaches aren't worth taking when trying to treat rare diseases such as these epilepsies, but it's why we're very much going down the small molecule route as a way to kind of have a series of adjunct therapies that we could potentially use to treat this form of epilepsy. So I'm talking about identifying KNA 1.1 inhibitors. What kind of inhibitors already exist? So prior to 2020, we primarily had 3 known KNA 1.1 inhibitors. So quinidine, which I mentioned before, and 2 other antiarrhythmic agents. So clofilium, which has a similar kind of efficacy as quinidine, so not ideal, and bepredil, which has an IC50 in a bit of a lower range, but also quite significant side effects. So not a significant amount of trials with this compound. Come 2020, a couple of papers come out, one of which was from our lab. So Beth Cole and her work, which I will talk about a little bit more in a moment, used a structure-based approach to identify a series of novel KNA 1.1 inhibitors. And so did a group from Vanderbilt University, David Weaver, who pops up a lot in this literature, quite a prolific discoverer of inhibitors. Following this, over the next few years, we had a few papers come out, a few different series of compounds. So the interest is gaining in identifying compounds that can block this channel. So it shows that we've got an appetite collectively for finding compounds that can modulate the activity of this type of channel. To the extent where Praxis Therapeutics in 2021, they announced that they were going to develop some of their small molecules for the treatment of this epilepsy specifically. They've been quiet for a couple of years, which could mean that they're either doing an amazing job and they've found a drug, or that they've just quietly dropped it by the wayside. If anyone knows, please let me know. I'm excited to find out. But in the interim, because we don't believe in putting all our eggs in one basket, we're storming ahead with our small molecule discovery efforts. So with a view to this, I'm going to build on how I mentioned before with Beth's work. So the questions that we want to answer using these structure-based approaches and this work is whether we can identify novel potassium ion channel inhibitors which are potent, so more effective than the ones we've got already, specific, we're not hitting hERG, amongst other ion channels, but please not hERG, and drug-like, so have therapeutic properties that can actually help us move any hits forward into a therapeutic space. So where did this start? So Dr. Beth Cole, who during her PhD did a lovely side project where she used a structure-based approach to identify some novel inhibitors. So she did a screen of around 100,000 compounds in silico, tested in vitro about 20 compounds, and she found 6 inhibitors of this channel, which is really lovely, both because we have some novel inhibitors now, but also because it shows that this protein especially is really susceptible to using the structure-based approach. The question being, can we scale this up? So this is 100,000 compounds. You could kind of do this on your local PC if you've got a decent PC, but we want to push this up into the millions of compounds. So what does a structure-based approach look like? So first up, our virtual high throughput screening of compound libraries. This can be hundreds of millions of compounds depending on what our computational power is. And then using our computational programming to dock against a structure. Fortunately for us, we have our cryo-electron microscopy structure, so that's a lovely target for us to dock against. So we then give us our scores so we can kind of prioritize which compounds we want to look at in vitro. Biosynthesize them. In the case of this project, we have been purchasing them from commercially available sources because I'm not a chemist and I can't have a crack at making them, sadly. So the ones that we can buy off the shelf, it is. And then we can biologically test them in vitro in the lab using a combination of different assays. And this is an iterative process. So the information that we get from our biological test, any hits that we get from our screen, we can feed back into our virtual screens and try and find things that are structurally related, but even more potent. And we can keep going around this process until we've found something that has that kind of like legendary, fabled nanomolar affinity against our target. So the first step, identifying our compound libraries. Which do we use? So when we use this approach, we're very much led by our resources and our overall aims. So the libraries that we've been using in this project, initially we use our in-house library. Here at Leeds, we have an in-house library of around 25,000 compounds run by the Medicinal Chemistry and Chemical Biology Group, the MCECB. And this is a really nice initial screen that we can use if we just need to do a bit of a proof of concept, or we just want to expand our pool because they're nice and structurally diverse. We can run these against our target, and it's a nice kind of first point of call. We then wanted to expand to some different commercially available libraries. So we have the enamine drug-like library, which comprises around 2 million compounds, and then Atava and Life Chemicals, all of which are quite structurally diverse. We've got a nice pool to work from when we're doing our screen. So we can get all these together and screen them against our structure. So once we've identified our libraries, we need to do the screen. But where do we dock? This is the biggest potassium ion channel subunit. So there's a whole lot of it to screen against. So there's a couple of approaches we can take. We can either use in silico prediction. So here on the screen, I have just a screenshot of a sitemap from a program called Maestro that we use, where it can predict based on the kind of available space in the protein where the most likely inhibitor binding or compound binding site is. We can also use known structural information. which fortunately for us, Beth had already done for me. She's really doing all the legwork in this project. So we have our cryo-electron microscopy structure. So this is the chicken KNA 1.1 again. And Beth did some docking of some known inhibitors. So these are bepredil and quinidine, the two antiarrhythmic agents that I spoke about before. And she docked against the pore region because it's been hypothesized that this is where these two inhibitors bind. And she identified two amino acid residues, which, point back up, okay, two residues which seem kind of pivotal in coordinating the binding of both of these inhibitors. So this T314 and F346. And off the back of this, she mutated them. And there's a bit of extra stuff going on down here, but basically the takeaway is that if you mutate these residues, the inhibitors have significantly lower efficacy at blocking our channels, providing a good amount of evidence that these compounds do bind to this site, and that this is therefore a viable binding target for any virtual screens that we want to run. So using this information that we have, we took this site, we put it into our virtual screen. So when we carry out our virtual screen, we have our ARC screening workflow. So the ARC facility at the University of Leeds is a high-performance computing facility that is available to people at the university. And we can use this to run some larger screens. So if I'm doing tens of thousands of compounds, I can do that on my PC, okay. But if we're doing millions, we need a bit more computing power. So what this kind of screen looks like if you run through ARC. So we can start off, say, for example, if we're running our enamine library. So the enamine library, they have approximately 2 million compounds in their drug-like database. So what we need to do first is kind of like narrow down the compounds of interest and kind of rule out the ones that are definitely not going to have any effect. So we use a more rapid program such as Fred, which is part of the OpenEye suite of software, which can give us a bit more rapid but slightly less high fidelity kind of understanding of where our compounds combined. And we can use this to kind of ration off the top 10 predicted binding compounds and take that forward to a bit more of a more thorough program. So in this sense, we use Glide. So Glide is from the Schrodinger platform suite of softwares, and we use their standard precision mode to dock the top 10% from our Fred screen against that site once again, just to get an idea of which compounds are predicted to bind the best. This is where the human component comes in. So it's all well and good getting a whole load of numbers and a whole load of chemical structures. Some of those chemical structures might say interfere with an assay. We have our pan-assay interference structures, which will, for one reason or not, whether it's fluorescent or if it causes some kind of cell death, will give you a positive response, even if it's not actually working properly in your assay. So this is where I will go to my friendly neighborhood chemist, Katie Simmons, my PI, and I'll say, hi, which of these is going to murder a person? And she'll say, don't choose this one, please. And I'm like, okay, cool. And we'll narrow it down from the 100 or so top scoring compounds down to a manageable and, importantly, affordable number of compounds, which we will then order. And because we've gone through our commercially available library rather than anything that might need to be synthesized, this is very manageable for us. So we'll buy those in, ready for the next step. So once we've done our docking, we have our candidate compounds and we've purchased them. What do we do next? So we need to evaluate these biologically. So what we've done is in-house produced a heteromeric cell line. So this is a HEC293 cell line, which expresses a concatemer of the wild type and a gain of function mutant of KNA 1.1 subunit. The reason for this being is that in vivo, in people who have these kinds of epilepsy, they won't have a homomer of the disease-associated mutant. So when we're screening, we want to make sure that we're replicating the disease, the condition in the disease state as much as possible. It also means that when we're running our assays, so say a fluorescence assay, because this mutation, so in our case, Y796H, it's such a gain of function mutant, we don't also have to include any kind of activator in our assays. For example, like loxapine, which we can use to activate the wild type channel, just to give us a bit of a higher response. If we're including activators and inhibitors in the same assay, that could cause some kind of issue when we're getting a readout. So we have this system as kind of a way to make sure that we can screen against disease-associated mutants, but also that we can get a good readout for our, for example, our fluorescence assays. So we take this HEC293 cell line and we take it forward into our assays. So we have a kind of standard in vitro compound screening workflow at this point, which starts off with a thallium flux assay. So we load our cells with a thallium sensitive dye, so we can use thallium as a surrogate ion for potassium in these assays. We use Thalos dyes. So when we'll load our cells with our dye, and here we go, our cells are lovely and loaded up. We'll add our thallium sulfate solution in after 30 seconds, and then we can quantify the fluorescence response in response to the addition of that thallium influx. And we can kind of quantify the slope of these lines, which correlate to the amount of potassium ion channels that are open or inhibited by our compounds. So this is just kind of an example of when we're screening these compounds. So these are a good chunk of the first few that we screened. These are all screened at 10 micromolar compared to Bepredil, which is at 30 micromolar. So we're looking to see whether this, in the first instance, whether we can have inhibition that's kind of comparable to that of our positive control, which is a much higher concentration than we want to see in our compounds. And thankfully, we get a decent hit rate. We tend to see around 15-20%, depending on which compound pool we're using. And it gives us a really nice indication of which compounds are worth taking forward in the first instance for future in vitro characterization. So once we've got an idea of which of our compounds are interesting to look at, we can kind of characterize them further. So we can use manual patch clamp just to make sure that we're not getting any kind of fluorescence artifacts when we look at our thallium flux assay. These channels are weakly voltage gated, so we can use a pulse protocol to stimulate them and then just perfuse our compounds just to see what kind of inhibition we're getting. And we can also look at our specificity. So this is one of the things that we really need to concentrate on, because obviously that's a big problem with all the other antiarrhythmic agents that we're looking at. So we've got a combination of fluorescence and patch-based assays that we have in the lab that we can use. So again, like the thallium flux assays for potassium channels, patching for hERG we've been using, and we're currently in the process of developing a automated patch assay with some cell lines using our patch liner that we've got in the lab. So using this workflow, we've tested 112 compounds in total. I have heavily obfuscated the structures of these compounds because we want to apply for a lot more grants, and if I tell you all of them now, they're going to be on the internet, so sorry, but trust me, they're really great. So we have a lot of structural diversity amongst our hits, and we've screened at things that have screened out everything that's got an IC50 over 10 micromolar, because that's the kind of cutoff that we have. And we've got a lot of hit matter to work with. So of our 112 compounds that we tested using this series of assays, we get a hit rate of around 17%. So we have 19 compounds with the IC50 under 10 micromolar. And that's really fantastic. It's a lovely place to start. We're really thrilled when we've managed this. But a couple of extra questions. For one, can we improve their drug-like properties? A few of these compounds had common denominators of things that we'd like to change. So for example, solubility of some of these compounds was a real problem. They would be effective at our channel, but not particularly soluble, which makes them difficult to test in different assays, especially if you're trying to do some kind of ADME testing. But also, potentially, we could be really losing out on some of the efficacy. This could be a nanomolar compound if it was just more soluble. So that's these sort of things that we want to look at. And also, is there a barrier to hitting this nanomolar thresholds? We know that a couple of the compounds published, so the Praxis compounds that I mentioned before, they've managed to get that nanomolar efficacy of their inhibitors of the channel, whereas these are all in the low micromolar range, which is great. And because the bar has been set so low by our antiarrhythmic reagents, it doesn't mean that if they were to be used as therapeutic compounds, they would be the worst. But we want to aim for the peak therapeutic compound. So how do we optimize this? So first up, we use more in silico tools. So we are big fans in our group of using as many computational tools to streamline our workflow as much as possible to ensure that we're getting the best out of the resources that we have. So we have been working for the past year or so with a company called Medchemica, who have a piece of software called MC Pairs. And MC Pairs is fantastic if you want to do any kind of compound optimization. And what you do is you can upload any kind of molecule. So I've kind of whacked in Bepredil as our example here. And you can tell it whether we want to increase or decrease a certain aspect of the chemical property. So for example, here we want to increase our solubility. And this uses a match pairing AI algorithm to determine whether any changes to a structure similar to yours have been made in previous literature and have had the desired effect. So for example, has it happened before? If you did this to your compound, would it therefore potentially do the same? And are there multiple instances of this happening? So we get something like this when we do this. We'll get suggestions for compounds that we could make or synthesize, hopefully by, and the impact this would have on certain different aspects. So not just the solubility, it will tell you if the, in previous instances, when they've made that chemical change to a particular structure, if that's also, for example, increased the amount of hERG liability that we see, or increased the interaction with other cardiac ion channels. So this is a really handy piece of software that we use. So at the minute, we're kind of prioritizing specifically solubility and hERG liability. And this is a real asset because now we have a PhD student who's started working on these compounds, and he is a chemist by training, which means that he's now able to synthesize some compounds using these suggestions from this match pairing algorithm, and hopefully enhance all these kind of aspects of the compounds that we've been struggling with. So this is where we integrate this in this part of it, in this iterative kind of process. We're getting our hits. We're then going through Medchemica, and it's giving us up to hundreds of compounds if we want to, suggestions that we could test, and we can screen this through our virtual screen to make sure that is still predicted to interact with our potassium channel. So that's fantastic. Second question that we had there is there a barrier to this nanomolar threshold? So we've been doing a lot of docking on the chicken cryo-electron microscopy, and that's been working great. We've been finding inhibitors that work really nicely. But we were wondering when we were talking after getting all these hits, why can't we get down just tenfold lower in terms of efficacy just to improve these compounds? Is there something that's going on from the kind of initial assumptions that we've been making that might be hindering us from breaching that threshold? So we thought we'd go back to brass tacks, look at the initial structural information that we have, and try and figure out what the situation might be. So this is the initial paper that we were working on. So this is the chicken cryo-electron microscopy structure. This group in 2017 published the structures in both open and closed states. So we have a couple of different structures that we've been working with. The human and chicken sequences have 84% sequence identity, which is fine for our purposes because the region that we've been docking against, that poor region, is fully conserved between the two species. So we have 100% identity between the human and chicken in that particular spot that we were looking at. But what if we weren't looking at 100% the right spot? Could this be throwing a slight bit of spanner in the works? So more recently, a potential kind of suggestion as to whether that might be the case kind of popped up. So this is a paper that was brought out last year And right at the end of when we were doing this work, I cannot convey to you how frustrating it is. We had done all this docking, then they bring this paper out. But this is the human cryo-electron microscopy structure. So it's a slightly higher resolution. It's of the human protein, and they've published structures in a few different states. So we have here, again, a few different kinds of slopes, a few different closed states in the red there, and a couple of different sodium activated potassium bound states. But also in purple there, we have a ligand bound state. And this is really exciting because the ligand that was bound to this protein in this structure is one of the praxis molecules from that paper earlier on, which I mentioned. And this is a really high affinity binding molecule in the low nanomolar IC50, which is exactly what we're hoping to emulate. So now we have an extra bit of structural information available to us where we can look and see, have we just missed something in terms of ligand binding? So when we interrogated this a little bit more, we found a little bit extra. So this is the initial cryo and electron microscopy structure of the chicken receptor. So you can see here, if I put my laser pointer back up, you can see here that where we've been docking, we're blocking the pore region quite nicely. It's also pretty central. So you're getting, from what we've been seeing without docking, you don't really have space for more than one molecule to kind of bind. However, when we look at our human structure, with our compound 23 bound to it, we can see that it's a bit more peripheral. It's kind of popping up between these S5 and S6 helix, and it's a bit more kind of to the side, kind of opening the possibility of having a few different compounds bound to this one structure. And if we kind of interrogate this a little bit closer, the reason for this becomes apparent. So we've been docking to our chicken structure on the right here, and our new structure, human structure, on the left. And what I've put here is the original phenylalanine 312 residue, as you see in the upper state in the chicken receptor, as we've been docking against. And this is compound 23 as found in the human structure, but in the chicken. So this is just to demonstrate how badly this compound clashes with this residue. So this side chain basically fully occludes any compounds kind of binding into this site. Whereas when we look at our ligand bound structure, this phenylalanine residue is flipped fully out the way. It's opened up this lovely little extra bit of binding pocket where our compound 23 is bound. And I was kind of beating myself up a little bit as to, I've been looking at different movement of residues. We know it's not a stationary thing. Why didn't I spot this? And I had to go at kind of using a couple of different computational methods to get this residue to move where it's been found in the structure. And you find that it's really difficult to force this residue to move to this region if you just have your structure. So if you ask Maestro or Pymol, which are the most likely kind of orientations for these side chains, it simply will not go here. You have to kind of like force it. So it was kind of an unexpected binding pocket that we have between these two helices. And when we have a look from the extracellular region, so kind of looking from up here downwards, we can see that this is our compound 23 in the human, kind of like sneaking off around to the corner between these two helices. But because it's kind of not as central as when we look at our docking in our apo state, which is fairly kind of like central in the pore, sitting quite nicely, but there's just going to be the one molecule there at any one time. You do open up the possibility that you could have multiple occupancy of our inhibitors in these sites, potentially kind of contributing to an increased efficacy of the compounds, or possibly just forcing it into a different conformational state. So obviously we took our compounds from our original docking and we looked to see whether this would make much difference to where our compounds were predicted to bind. So on the left here, we have our original chicken docking, and on the right, we have the human structure. So our original docking score for this compound, I hope my PIs aren't having a heart attack. These are the ones that are coming in the preprint that we're going to bring out soon. So, steal them if you like. So originally we have our docking here kind of nestling nicely in the pore domain. This 3-ene residue predicted to form a hydrogen bond. And that's the main kind of specific interaction that you're seeing between this compound and the structure. However, in contrast, when we dock to our ligand-bound human structure, we see because we've moved this phenylalanine residue flipped out the way and opened up this basically new kind of like binding spot that the compound can venture into. This is preferentially venturing into this pocket here. And we're also seeing an increase in the specific interactions being produced between this compound and our side chains. So we have a hydrogen bond between this phenylalanine residue and pi stacking interaction formed down here. And with that, we see an associated improvements of our predicted docking score, which is more within the region of what we would expect from the efficacy that we've seen from this compound, which is in the region of around 4 micromolar. So this kind of explains to a certain extent why this compound might be a bit more effective than we were expecting from our initial docking. And is also the case with the second of our compounds. I won't go through all of 19, don't worry, just these 2 examples, but just wanted to explore with this one. So again, relatively low docking score. We see predicted pi stacking interaction, these phenylalanine residues, but the bulk of this compound basically just kind of forced to kind of hang around in the pore region. Whereas when we flip this side chain out the way, again, as we see with a good chunk of our compounds, this is preferentially kind of like moving up and docking into this region between the S5 and S6 helix. Although interestingly, we are getting a loss of a specific interaction in this docking. It could be the case that it's just more of the compound is having some kind of interaction with the proteins. So, for example, we have a good chunk of our amino acids just kind of a good distance away from our compound here, which could mean that we have a less predicted affinity for this compound. And here, because it's slightly nestled between our 2 helices, we're seeing a bit more of an increase in predicted docking score. Either way, it's just very interesting to see. And when we dock our compounds, the kind of working theory is that we're getting the majority of our compounds docking to this site. Obviously, it's not a static image, it could be a mixture of the 2. So, which raises some future questions, including whether we can improve the potency of our compounds using these new structures, and also whether we can learn more about the binding modes of our inhibitors by using these structures. And off the back of this, whether molecular dynamics studies would help in our efforts. So we have a colleague in our lab, Dr. Eleanor Mitchell, who's been doing some molecular dynamics with KNA 1.1 and is looking at, well, primarily the sodium binding sites, but I'm hoping she can be coaxed over to the dark side to look at the interactions between some of our molecules with the KNA 1.1 structure, because looking at that kind of fluidity of structure, as well as in addition to these 2 kind of snapshots that we have, could be quite interesting. We also have our other structures. So that human paper did also bring out a few different closed states, a few different open states. So there's a whole lot to interrogate in terms of structural information and ligand binding. So a lot more studies to do. So if someone would like to fund me to do that, I would love that so much. So we didn't have time to run the whole screen again because this happened right towards the end of this project. So sadly, we couldn't go through the whole large-scale screen, in vitro testing, and so on with this new structure, which is a real shame. However, we did have time to do a quick repurposing screen. And the reason for that is because just the pool is a bit smaller than just all of chemical space versus all the drugs that we have. So I'm just going to, as a quick addendum, I'm just going to talk briefly on that. So drug repurposing in KCNT1 associated epilepsies, as with all diseases, but specifically with these rare diseases where funding can sometimes be at a premium, and there's quite an urgent need with how ill these children are. Drug repurposing can basically skip a few of the early stages of drug discovery when in comparison to identifying a series of novel small molecules, which have been completely untested in humans. There's an example recently, earlier this year, where fluoxetine has been shown to be a KNA 1.1 potassium ion channel inhibitor, so the antidepressants. And then this is an N of 1 trial, and the patient in this study found that their epileptic seizures were significantly reduced by treatment with fluoxetine. So there's a real appetite for drug repurposing, specifically particularly with this kind of epilepsy and the charities who are associated with them. So what we can do is run a quick drug bank screen. So the drug bank is an online repository of all the known drugs and particularly FDA approved drugs. And because this is a smaller pool, this is quite easy for us to run. I can just run this on my local PC. So we have 7,800 drug entries in total, of which around 2,200 are FDA approved small molecule drugs. So these drug entries also include peptides, which we tend to rule out, some illicit substances, which, if that's your flavor, I'm not going to stop you. But we tend to focus on the FDA approved drugs just for this kind of instance. So we run our screen as we did before. So taking the in silico library, docking against our structure, getting our scores. So here we got some really favorable docking scores. So here are the top 10 scores that we got from these compounds once we'd screened out all the peptides. So yeah, -10, that's kind of predicting kind of nanomolar efficacy, which is what we really want to see with these screens. And once we put these in, so this bar chart here is analyzing the inhibition of these compounds versus our positive control, Bepredil. So we saw from this 10, we have 2 that have some promising activity at 10 micromolar compared to our Bepredil control at 30 micromolar. So we took this forward and initially screening with our thallium flux assay because we were kind of short on time. So I still need to patch these. This is pretty fresh info. But takeaway message being that we've got a couple of hits, 20% success rate in line with what we were seeing before. And again, low micromolar efficacy of our compounds. I'm not going to tell you what they are for the time being. Hopefully we've applied for some funding to kind of move this a bit further forward. But one of them we know crosses the blood-brain barrier because it's a medication of that nature. So we think really promising. If we can scale this up against this new structure, we can really kind of unveil a wealth of information about potential future drug repurposing studies. So this new structural information, is, I think, not just drug repurposing, but also novel small molecules. We can really push forward with all this wealth of information that we have. So to summarize, KNA 1.1 is becoming more and more of a desirable target, especially now we have pharmaceutical companies getting involved and getting interested in these kind of epilepsies. And now that companies have a bit more of an appetite for investing in rare disease research. Of our compounds, we had a really nice hit rate. So around 17% of our 112 tested, resulting in 19 compounds that we have with an IC50 of less than 10 micromolar. A couple of our lead series we're taking forward, hopefully to develop further as a part of a hit to lead stage to develop as some part of kind of therapeutic treatment in the future. There is so much structural information for us to explore, and I would love to be able to spend several years just solely playing with structures and looking at the interaction with compounds. If you'd like to pay me to do that, please drop me an email. But yeah, it's really fascinating, and I think there's still so much to probe just in this very small area. So there's a whole ton of stuff. I'm sure very exciting research will be coming out of this in the next few years. And the big kind of take-home message that I want to kind of like put forward is that the value of a structure-based approach. We have a plethora of structural information when it comes to not just ion channels, also other proteins, but primarily ion channels because they're the important ones, right? So especially because we have things like AlphaFold now. So even if we don't have a experimentally derived structure, you're never truly 100% away from having some structural information that you can use to identify small molecule modulators or explore interactions between compounds and your protein. It's really, especially if you have some kind of like quite restricted budget, or you need to do something relatively quickly, or you don't have the capacity to do the incredibly high throughput that you might find in a pharmaceutical company. It's really worth considering. So yeah, if it's something that sounds interesting to you, please drop me a line, or anyone else I'm about to talk about on these next slides, because it's something that we're really trying to drive forward, especially in Leeds, because it's such a great approach. So that's all I have to talk about today. So I would just like to acknowledge some people. So Doctors Lippiat, Simmons, and Munch, they're the people who were spearheading this project, and without their expertise, it could not have happened. And the labs as well, particularly Rebecca Motley and Eleanor Mitchell, who are my partners in crime when it comes to KNA 1.1. Action Medical Research for funding this work in the first place. And please do check out Paddy's story if you can. It's a really well done video, and I think it really hammers home the message for why we need to invest in rare disease research. And please do get in touch with me if any of this sounds interesting. I'd love to have a chat about this stuff. All right, well, thank you so much, and I'm happy to take any questions if you have any.
Right, thank you so much, Emily. That was great. There's quite a lot of questions in the chat already, so we better get going. So the first one is from Raj. Considering that sodium activated potassium channels, KNAs, are opened by high cytoplasmic sodium concentrations and play a crucial role in regulating neuronal firing patterns, how can we experimentally obtain the open channel structure of KNA without applying voltage? So kind of a structural question. Do you know how they got the open?
Oh, yes. So there's so that second paper, they kind of produced some kind of it was kind of inside out vesicles so that you could have the structure and you could change the amount of sodium that you could have kind of applying to the intracellular face so that you can kind of artificially open the structure without having to kind of apply a stimulus. So that's how, and then through the cryo-electron microscopy, had a few different kind of titers. So which is why we had in that second paper like 6 different structures, because they have kind of organized the classes so that we have different kind of like substates within the kind of apo and open states. And also applying different concentrations of sodium just to check, just to see whether there was any kind of like structural differentiation between the different concentrations. So it was really cool.
Okay. So I think this one was posted before you mentioned it in your slides. How similar is the chicken KNCT1 to the human? I think you said 80% or?
Yes, I can slightly elaborate on that as well, because it's something that I didn't put in my slide. So there have been a couple of compounds that we were interested in, which they showed kind of species disparity in activity between the human and the mouse in this particular case. And we were trying to figure out for ages why that was the case. And because we were looking at the pore region, we were thinking, this is where these things bind, and it was just fully conserved between the 2 proteins. But hang on, if I can get my, sorry, I'm just going to zip back real quick so I can point at a structure. Here we go. So in the map between the mouse and the human, we have a couple of amino acid residues here, which are different between the human and the mouse, and they were shown to kind of help in the mouse slightly push the compound out of this binding site a little bit, which to a certain degree can explain why you're seeing this kind of species disparity, because if these compounds are binding here, then this is the region where you're seeing some structural difference. And I think in the chicken as well, although we haven't tested our compounds against the chicken receptor.
Okay, so the next one is from David Colhoun. If a conformational change is involved, and I think it is, a change in structure anywhere in the molecule will affect binding. So maybe you can, I think you kind of touched upon that, but maybe you can elaborate. He would also link to, I think, his recent paper that explains the difference between efficacy and affinity of binders.
Yes. So is the question, does that affect?
Yeah, I guess depending on the structures you start with. I know you kind of commented that towards the end of the talk. So how would that affect binding?
Yes, for sure. So this is why, because even though we're primarily kind of focusing on these sites up here, we are also bearing in mind other more kind of peripheral sites. Like, so for example, the sodium binding site is one that we're really interested in because it shows relatively, from what we can see, relatively small changes compared to when we're kind of looking between the open and closed states around the kind of transmembrane region here. We're seeing a bit more movement, a bit more potential displacement. So it could be the site that we're docking to, that could be feeding into the relatively low affinity that we're seeing for our compounds. So yeah, it's definitely something that we're bearing in mind when we're kind of exploring more docking sites and trying to kind of improve our approach using the structure.
The next question is from Damien Bell. Lovely work, Dr. Caseley. Thanks. Does the thallium flux screen allow you to find openers? If yes, did you find any?
Yes. So interestingly, you can find openers. Very interestingly, in the screen that we ran with the random small molecules, the novel small molecules, we didn't see any activators or anything that activated to a significant degree. But when we did the drug repurposing screen, one of the compounds that we looked at was doxepine, which is, because I was going in just kind of ordering things randomly, a structural analog of loxapine, and that was an activator, just nowhere near as strong as loxapine is. So yeah, so we can identify them, and it's just kind of, I would love to dig into why we didn't find a huge amount of them when we were looking at our random small molecules. It's a really interesting point.
It's another question is from David Kohonigan. What is the correlation between the predicted potence and that observed, insofar as the latter can provide an estimate of equilibrium constants for binding?
So in terms of docking scores, we generally tend to be in the kind of general ballpark, but it's very much taking it with a pinch of salt. So for example, with some of those compounds in the drug repurposing screen, we're seeing scores, predicted docking scores of -10, which you'd think would be kind of low nanomolar range, broadly affinity. But it was about 100-fold out when we do the actual screen. So this is very much, I would say, an imperfect science. It's very much a tool to kind of get you towards where you want to be. But as these tools are refined and improved, we're really hoping that the information that we're getting from studies like these, we can feed back into these kinds of algorithms and help them to improve, especially when it comes to targets which aren't as well known as, say, for example, hERG. There's so much structural information about hERG and ligand binding, but if we can use studies like this to kind of feed back into any kind of like machine learning based tools that we have, then yeah, I think it's a really valuable thing to do.
Did you maybe notice any sort of bias? Does docking consistently underestimate or overestimate compared to the experiment, or is it all over the place?
I would say it's more likely to overestimate, but also that could just be a bit of bias because we tend to only buy in the ones that have particularly good docking scores, whereas the ones with lower docking scores, we tend to kind of rule out. So there could be some down there with poorer docking scores, which could be fantastic inhibitors, but we haven't even looked at them because we're trying to prioritize the ones that we think will find the best. So yeah, that could be a bit of a confirmation bias thing on my part.
And David also asked, can you estimate the extent to which the channel needs to be open for block to happen?
Oh, that's a good question.
Yeah.
So we docked the apostate. So if this is at all kind of representative of what's happening when our compounds are binding, I know I've just spent a long time talking about how that might not actually be the case with this new ligand structure. But I would say not particularly to a great degree. I think you can target this apostate rather than the open state kind of preferentially. But that is a guess.
So from Kim Lacan, thank you for your talk. You mentioned that Kv channel to be expressed in sensory neurons as well. Is it involved in painful stimulus transmission?
Less so much pain, but itch is a big one. So activators of this channel have recently been used in mouse studies, in mouse models, to show that you can target chronic itch. So that's why if we do find activators, that's also quite interesting because we have something that we can use as well as epilepsy treatments. There's kind of things in the opposite direction that we can look
at. So David also asked then, have you ever looked at the effect of your blockers on single channel currents?
I haven't, no. I would really like to though. It's very much top of the list. It's been wholesale so far, sadly.
Yeah, well, high throughput.
Well, yeah, exactly.
Mark Rogers, lovely talk, Emily, from an old fan of KNA channels. Have you or will you test any of the competitor small molecules to benchmark your assays and drug screening efforts?
Yes, we do have we don't know which of the Praxis molecules they're looking at in their therapeutic kind of trials, if it's any of the ones that they've published, but we do have one of those molecules that we can compare to, because obviously we do want to go, we don't want to just be okay, we want to kind of be pushing the boundaries of what we can achieve. So we have that, especially when we have our assays, which are not quite 100% tallying with assays that other companies have used, because we have this heteromeric HEC293 cell line. We want to make sure that our compounds, the readouts that we're getting, are comparative to the ones that they're reporting in their publications. So yeah, we've been using that as one of our positive controls for the time being.
So the next question kind of builds on the chimeras that you use. What is the current amplitude of the chimeric channel as compared to the wild type?
So with the chimera, a couple of nanoamps you'll see maximum. In the wild type, like a couple hundred picoamps. It's several fold change. It's pretty huge.
And did the intracellular sodium concentration influence compound IC50?
To be honest, we haven't actually, well, because there's a preprint out, I'm not sure if the full publication has come out yet, from Beth Cole again, who was looking at different mutants and their influence on sodium activation of KNA channels. And she found that there are some gain of function mutations in KCNT1 associated epilepsy, which just allow the channels to open in the absence of sodium completely. So in terms of gain of function mutations, the sodium might not be quite as much of a pivotal part of it as if we were just looking at, say, wild type channels.
And targeting the pore region, isn't it quite conserved across potassium channels?
Yeah, I think that's why we're having so many hERG problems, which is why, yeah, it's one of these where it's, in the first instance, finding something that we know is going to inhibit the channel. But also it's why we've kind of tried to diversify our chemical space a bit. So, for example, trying to push towards some larger compounds, possibly because the pore region of this channel is a bit larger than, say, hERG. We can maybe try and kind of like force out the possibility of inhibiting hERG just through sheer volume. But obviously, in terms of a drug discovery perspective, that's not ideal. So we are also looking at, so with MC pairs software, with the match pairing, there are kind of some known structural changes you can make to compounds which help to reduce the inhibition of hERG. And there's a lot of information that these match pairs software is trained on about hERG because it's so well characterized. We are also looking at investigating different sites. So, for example, like I was saying, with the sodium binding site that my colleague Eleanor is looking into, this seems like it seems a bit route one, but also a very a binding site that has a lot of potential. So it's a case of taking these, improving the compounds that we have, and investigating further opportunities now that we've got more structural information.
Okay, so the next question is from Damien, who is trying to justify that energetically unfavorable position of the phenylalanine 3,1,2 side chain. Is that due to the ligand binding in ligand bound state, or is it possible that it's an artifact, cryo-EM artifact?
So I got our resident cryo-EM professional Stephen Munch to look at this, because we were wondering about this when we first saw it, because it did seem a bit kind of far-fetched, but he was pretty confident that it's not an artifact and that the density that you see in the cryo-EM map is kind of representative of the orientation that they've kind of populated this residue here. So I'm pretty confident that it's because this ligand is binding and it's kind of forcing a movement of that side chain.
Okay.
Yeah.
So not an artifact. Okay, so we have 4 more questions. If you don't mind, I will continue for another few minutes.
Okay, go for it.
People are still hanging on, so if you don't
That's
mind,
very nice of them.
we might as well. Juan Pina Crespo is asking, have you tested any of your candidate compounds in native channels in human neurons?
No, we haven't. That's the next step. So in our lab, we have a mouse model of this kind of epilepsy, but we sadly don't have any kind of primary neurons. And that would be something that I'd really love to test, because obviously the human system is the kind of pinnacle of what you want to test these compounds against. So if there's anyone out there who has some neurons and they want to share or test these compounds, please let me know. I'd be thrilled to have a look.
Okay, so the next one is from Guy Salama, who is wondering, is there a way to kind of go beyond the pore region? So there is this tacit assumption that the compounds would best act on the pore region of the channel. What if the binding occurs in this large intracellular region of the channel? Are there more general approaches to consider such possible interactions beyond this?
Yeah. So when I was talking about the site mapping kind of function that we have, so the docking that we do is partly done in Glide, which is it's in a program called Maestro, and Maestro also has that site mapping function. So we can look at a number of different sites, and we can basically, if we want to, give it carte blanche to say, just ask it to every single site that anything could possibly dock to, and we can then go through. Sadly, with this program, we can't kind of dock against the whole protein because it's so big, but also because it's a tetramer, we can kind of narrow down the region that we're docking against because we get fourfold symmetry. So we can kind of look at like a subsection of our receptor. So I would say, yes, it's definitely worth investigating different sites. We're very excited about a few different sites to look at, especially including these sodium binding sites. And I would love to be able to investigate where different compounds might bind. But in terms of our candidates, I've had a couple of look at a couple of other sites that have been predicted from that site mapping function, and overwhelmingly they do preferentially bind in this pore region.
Okay. Great talk. Thank you. Did fluoxetine, other antidepressants appear in your repurposing talk results?
Fluoxetine was quite high in there, yes.
Yeah, okay.
So that was a really exciting kind of proof of concept.
Okay, so sorry, did you want to?
Oh no, I was just going to say, so that was really exciting, but yeah, that wasn't helpful.
So David Cohen says, nice work, thank you, but has one last question. Perhaps it's not surprising that docking scores are so often imperfect. Many of the forces involved vary as r to the power of 6. So changes in structure that are beyond the resolution of structural studies may be very important and yet difficult to capture, right?
Yes, definitely.
So there's always that aspect of this. Okay, that is great, Emily. I think that's all the questions in the chat, and because we are over time already, I will leave it there. Thank you so much again.
Oh, well, thank you so much. I really enjoyed it.
Yeah.
I will. Thanks. Bye.
Bye.
Thank you,
Thank you,
Emily.
Emily.
Bye.
Bye.
you.
Thanks.